Thank you for being here this morning to listen to our closing keynote and another day of great programming. Thank you all for the tremendous feedback that we have gotten already about the conference. It's heartening to hear that this is a program that is meaningful to you all, and it certainly is meaningful to us. So it's my pleasure to introduce our closing keynote speaker, Dr. Carol Feghali Bostwick, who is the Kitty Trask Holt Endowed Chair and Professor of Medicine in the Division of Rheumatology and Immunology at the Medical University of South Carolina. I got through the big words first, you know? It's like I ran out of breath. I can't begin to say enough about Dr. Feghali Bostwick. She is a tremendous mentor to all of us in the organization. She is the vice chair of our National Board of Directors and the chair of the board's research committee. She is a leader in her field, a recognized tremendous colleague and leader in scleroderma research, but most importantly to the foundation, she is a person who never says no, goes beyond every request that we could ever make of her. She is always available to us and always available to you. I see her around the country at chapter events every now and again. It's tremendous what she gives, and she is here today to give us a tremendous closing keynote that I think you will find absolutely amazing. It is such a fun presentation. And to my great mentor and colleague, I want to welcome Carol Feghali Bostwick to the stage. Thank you for being here, Carol. You can get to where you need to
Yep, thank you. Good morning, everyone. While I pull up my PowerPoint presentation, it's great to see you all. It's always so wonderful to also see how many people are first-time attendees. I also very much enjoy seeing the younger crowd. So let me ask you, how many people have watched my talk on YouTube? How many people know what's coming up after my talk? All right, good news, bad news. We are having Jeopardy. Bad news, the questions and the answers are new, so you're going to have to pay attention again. All right, so we're going to talk about research in scleroderma. We're going to talk about how cool science is. We're going to talk how most of the time some of the most interesting discoveries in science are accidental, because that is really truly the case. But it also makes a case for why it is important to support research in general. So for those of you who may not know me, I have been doing research on scleroderma for a long time. I've been doing research on scleroderma for more than 20 years. I, of course, started when I was 2 years old, but so I've had some experience in the field, and I wanted to talk to you about why research is important. Research is important because, to put it in James Adams's word, the great use of life is to spend it on something that will outlast it. And that's one of the main reasons why scientists do what they do, because they know that the impact that they might have, if they're fortunate that their research results in something, will last long beyond their own lifetime. And research is important because often the discoveries that we make as a result of research that's done in the laboratory at the bench are the discoveries that lead to therapies. So you often hear of the drugs that are being developed by a variety of companies, but what we don't often realize is the essential discovery, the basic discovery that led to the development of that drug happened in the laboratory. All right, and why is research on fibrosis and scleroderma specifically important? You know, you all have a role to play in this, and I think it's important to know why studying scleroderma and having funds to do research on it is critical. One of the hallmarks of scleroderma is fibrosis, and fibrosis is thickening. Thickening means increased collagen production, other matrix proteins that make the skin, the lungs, everything thicker. And fibrosis can affect any organ, but fibrosis is also an end result of many other diseases. So many of the diseases we know about, they may affect the liver, such as alcoholic cirrhosis or hepatitis C. The end result is liver fibrosis. Fibrosis can happen in the eye, in the kidneys, in the liver, in the GI tract, in the lungs, in the heart. It can affect any organ. And so understanding fibrosis and scleroderma will ultimately teach us a lot about all these other diseases out there whose end result is fibrosis. And actually, Thomas Wen at the National Institutes of Health determined a few years ago that fibrosis as a complication is responsible for 45% of all deaths in the developed world. Almost half of the deaths in developed countries are because of some form of fibrosis. So as I mentioned, fibrosis can affect any organ. This is a beautiful illustration by Linda and I that shows all the various organs that can be impacted by fibrosis, as well as the diseases that are represented. And this is another way of showing it. Basically, it shows why scleroderma research is important. Fibrosis in scleroderma can affect multiple organs as well. So scleroderma can serve as a prototypic disease to understand other diseases that also have fibrosis. And so when I hear someone say, well, why should we fund that much money for scleroderma research, or why should we publish papers on scleroderma research? It's such a rare disease. And I find that very short-sighted, because it may be a rare disease, but unlocking the mysteries of scleroderma and figuring out how to treat the disease will be applicable to a lot of other diseases out there. So it will have a very broad impact. All right. So I've always viewed scleroderma as a research puzzle, and I feel as we put the pieces together on the different manifestations and aspects of scleroderma, we can ultimately figure out what causes it, and most importantly, figure out how to develop a cure. And I'm going to teach you a little bit some of the lab terms to understand. And remember, there's a quiz at the end, so we're going to focus on the Jeopardy game on the facts, and as a hint, on quotes. So remember the quotes as well. All right, so in the laboratory, we do a lot of different assays to identify agents that cause fibrosis or other features of scleroderma, but also to identify agents that can stop it or reverse it, so both angles. The assays that are done, three main ones are done. Assays that are called in vitro assays, which means they're done in the laboratory. In vitro literally in Latin means in glass. So basically, if we grow, let's say, cells, and we put them in a plastic dish in the lab and add something to them that we think can increase collagen production, and then look at things that may stop that, that's called an in vitro assay. In vivo is when we do the experiments in a living body. Usually that's animal models, especially mice, more commonly used for scleroderma research than any other animal. So when it's in the living body, we call it in vivo. And when it's outside the living body, but still in somehow of a form that's similar to what's in the living body, we call it ex vivo. So ex vivo is outside the living body. So ex vivo, for example, is one of the assays I'm going to show you next. If we can take, let's say, a piece of skin and use it as is, as an organ in the laboratory, we call that ex vivo. So often our experiments in testing drugs involve in vitro assays, so in the laboratory, and they involve in vivo assays, usually in mice. But as you know, more than probably 95% of drugs that are tested in the laboratory and in animals fail when they get to the human clinical trials, because we are not mice. We don't respond like mice. And cells in a plastic dish are going to respond differently than our whole organism. So we developed a way several years ago to be able to test things and determine if they're likely to be effective in humans. And yes, I know you just had breakfast, but the good thing is I didn't show you this before breakfast. I showed it to you after. But this is human skin from plastic surgery, this specifically from a tummy tuck surgery. And the thick lower area that you see is fat. And several years ago, I wanted to know that one of the agents we were working on as a potential therapy would work in humans. I wanted to know beyond the mice that this is likely to be effective. So I decided to take human skin, clean up the fat, grow it, and test one of the factors that we had been working on called insulin-like growth factor binding protein 5, or IGFBP5, which we knew could cause fibrosis. And so you take the skin, the pink is the nutritious media, sort of like the laboratory equivalent to Ensure, if you will, and what the cells like to live in. And then You keep it going in the lab for a week or two, and then you look at what happens. And you can measure dermal thickness, just like as your doctor can sort of do the gentle pinch to determine how thick your skin is. We have ways of measuring how thick skin is in the lab. And so the way we often do it is as if you slice down a picture through the skin. So this is what it looks like if you slice through the skin. The top part is, this will work, I think, yeah. So this top part is the outer layer of skin exposed to the air, where you see the epidermis, and then as you go deep into the skin, inward. So we measure it this way. So if we take human skin and here inject this protein that we know causes fibrosis, I think without being scientists, you can see how thick it becomes compared to how it started out or before we injected it. So now we have ways of knowing that something that causes fibrosis also in human skin, not just in mice or in the test tube. So the way we've designed our research is we identify something that's abnormal in scleroderma. We then test it in vitro, remember, in glass in the laboratory. We test it in vivo in both the skin and the lungs of mice, because obviously lung involvement is also a very important problem in scleroderma. And then now, at least for my group, at the end, we tested in human skin ex vivo, that's maintained in organ culture, to know that it is more likely to be effective in humans when we get to the clinical trial. So with that in mind, we made a discovery a few years ago that endostatin, which is this last piece of collagen 18 that gets cut off from the rest of the molecule, had interesting effects in cells. So we had shown that this protein that we discovered causes fibrosis was increasing levels of endostatin. And our automatic reaction was then endostatin must be causing the fibrosis. It must be the mediator between this and the fibrosis. And so we wanted to test that. And obviously then we went to the in vitro assays and we took fibroblasts, or cells that were grown from the skin or lung tissues of patients, and we tested endostatin. And what we found, and you're going to learn to read science again, if you look at this signal, this is how we measure protein in the laboratory, and this is collagen. And the darker the signal, the darker the band, the wider and the darker it is, the more there is of that protein. And the thinner it becomes, the less there is of the protein. So what this shows you is this is a lot of collagen, and everywhere there is an E, that's endostatin, that shows you how much we were able to reduce the collagen with endostatin. So much to our surprise, it turns out that endostatin is not mediating the response to cause fibrosis, but it's trying to shut it down. And so that was our initial discovery, and we were very excited. Well, this is really me. I couldn't find a curly-haired, dark-haired scientist in clip art, so I had to somehow tweak it. So we were very excited, and that was the initial discovery. So obviously, one of my favorite quotes is Louis Pasteur, who said, "Chance only favors the prepared mind." Our mind was prepared to see that endostatin is one of the causes of fibrosis. And what we saw was the total opposite of that. But we were prepared to accept any data that would come out of our experiments. All right, so then we decided, well, endostatin as a therapy is cool. It's actually undergone phase 4 clinical trials in China for stopping cancer metastasis because it prevents new blood vessel formation, so the spread of cancers. But it was still very big for us and cumbersome to make and very expensive to make. So we decided to chop it down into pieces. Here's one piece, here's another piece, here's another piece. We chopped it down to try and figure out what is the smallest piece we can find that would still work to shut down fibrosis. And at the end, what we found is it was this piece that we've called E4. So we tested it in vivo. In vivo is a living organism. So this is mice in mouse skin. This is normal mouse skin. See how thick it is? This is mouse skin that you make fibrotic. You cause fibrosis. It becomes much thicker. And this is when we added our E4 peptide. So you can see it goes back to pretty much normal. So we were very happy, obviously, by that. We then tested again, but this time what we said, most patients show up to the clinic and they already have fibrosis. So it doesn't help me to be able to block it in a mouse. I want to know, can I change things back? Can I reverse it? So this time we caused the fibrosis in the mice, in the skin, and we waited. And after the fibrosis had started, then we treated. And this is normal skin mouse. This is when you cause the fibrosis, and now this is when you treat later. So clearly we could reverse this. So we could reverse the fibrosis, take things back to the way they were before the fibrosis. We obviously had to also change and do different ways. So we tried, one of the standard ways in scleroderma research is to cause fibrosis in mice using what's called bleomycin. Bleomycin is a chemotherapy. Years ago, they found that some of the patients who are getting it as a chemotherapy for cancer were also developing lung fibrosis. So scientists jumped on that observation and used it to induce experimental fibrosis in mice. So we also looked at the lungs in addition to the skin. And what we found is the darker the blue in the mice, this is bleomycin, the more collagen there is, the more fibrosis there is. And what we found is that with the peptide, whether we give it at the same time or later as a therapy, was effective at making the lungs look normal again. Then we went to our human skin. So fine, we can cure anything in mice. I can tell you, most scientists can cure anything in mice. They're the easiest to cure, but our thought was, is this likely to work in humans? So we took the human skin I showed you, we injected it, so this is how it is normally. We injected it with something to cause fibrosis, so now it becomes much thicker. And then we gave our peptide, and now it's almost back to what it was normally. Same if we waited, caused the fibrosis in human skin, waited a few days, and then gave our peptide. Again, we were able to reverse things, suggesting that there is a good chance that this will ultimately work in humans. So the key to treating fibrosis and scleroderma in general is not just to stop the progression. So I'm sure you've heard of drugs recently approved for idiopathic pulmonary fibrosis, which really reduce the progression of lung fibrosis. So our goal is not just to stop or reduce the progression. Our goal is to reverse the fibrosis. And one of the important ways to do that is to affect the extracellular matrix. Excuse me. So when we make a lot of collagen, what happens is the collagen goes outside the cells, and it forms these fibers. And then there's an enzyme called lysyl oxidase that then links these fibers to each other. And the more linkages that are formed, the stiffer and the more stable the matrix becomes, and the harder it is to break that down and remove it. So what we found is it turns out that E4 was reducing the amount of this enzyme. So what it's actually doing is it's reducing how much cross-linking is in the collagen fibers outside the cell and allowing the enzyme that degrade matrix to then break down the collagen and take it away. And that's how we're able to change things that were already started. So where are we now? We've actually been making it, the peptide in plants, because plants are very good for making small proteins and peptides in a very cost-effective way of making it. And it's interesting because the plant of choice is a plant in the tobacco family. So this is what this is. And yes, it's not done like you would in your raised bed or your backyard in your garden. This is a facility that actually produces it in large amounts in plants, and everything is automated and robotic. So that's the way that's happening. There are other things we're working on in research. So a lot of the investigators around the country have a variety of things in the pipeline that they're testing. One of the observations everybody has made over the years is, as you know, scleroderma is more common in women than in men. So we've always suspected that hormones may have something to do with it. And another observation we made several years ago was that, again, if you take this human skin and test it in human skin, if you inject estradiol, which is one of the estrogen hormones in the body, into human skin, you can cause the skin to thicken. And then if you use something that blocks the effects of estrogen, you can get the skin to go back to normal. So we're also working on trying to plan a clinical trial, try to get funding for it, to look at one of those estrogen blockers, and if that would improve things in scleroderma. And I'm sure the gentlemen who are patients in the room would say, well, what about us? Well, it turns out, believe it or not, that the estrogen levels in men with scleroderma are even higher than those in women. So we're looking also at changes that are on top of the DNA. So as you know, years ago we did a study on twins to see if scleroderma is an inherited disease. Turns out it's not directly an inherited disease, because of the more than 50 pairs of twins that we had in the study, we only had 3 pairs in which both had the disease. So that means it's not purely inherited, because otherwise you would expect all identical twins to have it. So now we're looking at things that are on top of the DNA. They're called methylation changes. They can be altered by diet and a lot of environmental factors we get exposed to. And we're comparing them in our twins, looking at the twin that has scleroderma and the twin that doesn't. We've identified actually a lot of interesting differences, and now we're in the process of trying to understand what those differences do and what they mean. So that's another one of the studies that we are doing in twins. Okay, so I want to, before I move on to some of the other examples of accidental discoveries in laboratories, I want to acknowledge my team, especially two very talented members, Yuki Yamaguchi and Logan Malakar, who've been amazing driving the E4 project forward. All right, so this is where we are in our research. So that's our science from my team. I want to go over some of the other accidental discoveries just to have more potential questions for our Jeopardy game too. And a lot of them are new stories, like I mentioned, in case you were prepared for this. So Plato said way back that science is nothing but perception. And I think that's a very interesting thing to say and something to keep in mind. And I wanted especially for the younger people in the audience to know that science, anyone can be a scientist. Scientists come in all sorts of sizes and shapes, all sorts of genders. You can be a happy scientist, you can be a mad scientist, you can be a woman, you can be a man. It takes all sorts of people to get science moving forward. All right, a lot of research discoveries that happened accidentally in the world have led to the development of a lot of the drugs that we currently use, from antibiotics to antidepressants, to a lot of the vaccines, even procedures and surgeries. So we're going to talk about them. We're going to start with an easy one. I see that way in the back. So Viagra is interesting. Viagra was initially developed as a drug by the company to be able to stop chest pain. So it was intended for people with angina, so for chest pain. And they did a clinical trial, and they distributed the drug, and it failed. It didn't work at improving chest pain. But then when they asked all the study participants to return the unused pills, no one would give them back. So when they looked into it further, then they realized that Viagra was effective for erectile dysfunction and ended up being a blockbuster with billions sold in just the first quarter. The discovery of a penicillin, a very important antibiotic, also happened accidentally. So Alexander Fleming was working on bacteria that grow in clusters like grapes called Staphylococcus. And he was going on vacation, so he had all the bacteria, which look like these small white spots. They grow in colonies like that in these Petri dishes. This is what Petri dishes look like. So he had the bacteria in these dishes, in these little colonies growing, and he went on vacation and left the dishes out on the bench in the lab. And when he came back, he found this mold or fungus growing on the dish. And he found that all the bacterial colonies that were growing around the fungus had died wherever the fungus was growing. And ultimately did the research to find out what was it that this mold was making to kill the bacteria, and discovered penicillin. All right, insulin is another story you might be familiar with. We've talked about it before. So this was in the laboratory when they were looking at the role of pancreas. So they removed the pancreas of a dog to know how would that affect the GI tract. And later, one of the lab assistants came and got the group and said, something is weird because there's all these flies swarming on the urine of the dog. So they looked at it, and they determined after they tested the urine that the urine had a lot of sugar in it. And that's how they determined that the pancreas was important. Ultimately, it led to the development of insulin and the availability of insulin for patients with diabetes. All right, infant jaundice is another one. This was a nurse in England who was very astute and noticed that all the preterm babies who had jaundice, who were put close to a window, close to the light, had the jaundice resolve fairly quickly. And that led to the discovery that actually light, natural light, causes the breakdown of bilirubin because the infant's skin is almost transparent at that stage. All right, substitute sugar, for those of you who use it. The very first one that was discovered was saccharin. There's been a bunch since then. it's interesting because I think the way we currently do research now in the bench, things are so regulated, and there are so many things you're allowed and not allowed to do. And I think if it were current policies, we would have never discovered substitute sugar. Because the way all these were discovered were by members of the lab either not washing their hands and eating food then that tasted sweet, or literally testing what's on their bench and tasting it. So accidental discoveries because of maybe what we call now sloppiness in the lab, but if they hadn't done that back then, and if the policies for safety were the way they are now, we would have never had substitute sugars, or at least not this fast. Pacemaker is another accidental discovery. So for the pacemaker, it was in the process, so this individual, Great Batch, was in the process of developing an oscillator. So he wanted to detect heart sound, heartbeats. And he put the wrong transistor in, and when he turned it on, realized that with that transistor in place, that oscillator was emitting a rhythm that was a lot like heartbeat, and that led to the development of the pacemaker, with the first one being put in humans in 1960. In terms of anesthesia, that was truly also accidental, although maybe more fun in developing it. So this was actually a result of laughing parties. So in the 1800s, they used to use laughing gas, ether and nitrous oxide, for recreational purposes. It was the laughing parties. And some astute individuals noticed, specifically one individual who was at the laughing party, had used the laughing gas, had injured his leg and was bleeding, but felt no pain. And ultimately, that led to the identification and the development of anesthesia, which has been, if it weren't for that, you can imagine how many of our current treatments would be literally a big pain. All right, the treatment for alcoholism, one of the first that they came up with was called antabuse. And the way that came about is there were two students in pharmacy in the Danish School of Pharmacy, and they were developing a drug for treating parasitic stomach infections. And like in those days, the first thing you do is you test it on yourself. So they took it, and then they went later that night to a friend's party and had a little too much to drink and felt very sick afterwards. So they realized it must have been because of what they had tried in the lab earlier, and that's how they developed actually the first drug for alcoholism. Blood thinners, often used in scleroderma as well, were developed also accidentally, although these were not laughing parties. This was actually a tragedy in cows. So this farmer in Minnesota suddenly noticed his cows were all dying, and they were hemorrhaging, they were bleeding, and he didn't know what it was. So he enlisted the help of a biochemist. And the biochemist thought that maybe the clover hay that the farmer was using had become rancid. So he tested everything that was in the clover hay and discovered warfarin, which is a blood thinner. And one of the last examples is something that's still in progress now, an accidental discovery where drugs that are commonly used for malaria have been determined to probably be effective for multiple myeloma. So a case of repurposing a drug used for something and now used for something else effectively as well. So, with accidental discoveries, it's key then to keep an open mind, to always be prepared to the unexpected. We always formulate a hypothesis in our research, and we sort of expect, we know what the anticipated results might be. And I think it's critical to also be prepared for something that's not anticipated. Joseph Henry said, "The seeds of great discoveries are constantly floating around us, but they only take root in minds well prepared to receive them." All right. How can you contribute to research? So obviously you have a role too, and multiple roles that you can play. John Richardson says that when it comes to the future, there are three kinds of people: those who let it happen, those who make it happen, and those who wonder what happened. And the fact that you came to this conference says to me that you are part of the second group, those who make it happen. So how do you make it happen? Well, activism is critical for any discoveries to make it to the clinic. And activism has resulted in a lot of things historically. Increase in federal funding as a result of activism and advocacy and your voices being heard, compassionate access to drugs, reducing the cost of expensive drugs. There's actually cases where patient groups go forward and put pressure on the companies to reduce the cost of drugs that are too expensive, repurposing of drugs, but also accelerating the approval of drugs through the FDA. When a drug shows a hint that it might be effective, then your voices will be heard if you speak up. So your voice carries a lot of weight. Don't underestimate what you can do if you speak up. Another way you can make a difference is through supporting research. As you know, the Scleroderma Foundation has a research grant program. We accept applications every year on September 15th. They're assigned to a scientific review panel, a panel of experts who then rank them based on scientific merit, and then the grants are issued. We have two general types of grants. One is for established investigators who are more senior with more experience. One is for the younger generation, the newer investigators. To date, we've funded, I know, way more than $25 million in research, and we've started a brand new research program called the Multi-Center Grant. This is a grant where we encourage more than 2 centers across the country to collaborate, or at least 2 centers, to collaborate and submit a grant. The grant is $500,000. We actually gave out 2 of them this past year for $1 million for this grant. The total funding of grants, of brand new grants this past year from the Scleroderma Foundation was $2 million, which is very impressive. The only other larger supporter of scleroderma research is the NIH, the National Institutes of Health, but in the more recent years, it hasn't been by all that much more than the Scleroderma Foundation. So if it weren't for the Scleroderma Foundation, I think the scleroderma research community would truly be struggling to be able to do research on scleroderma. And the last way, or one of the other ways that you can participate in research, is when you are asked for blood for a project, blood sample or skin punch biopsy. If you can participate, it's very helpful for our research. Like a small skin punch biopsy the size of the back of a pencil eraser is enough for us to grow cells like a carpet and use those cells for years in our research. So that's another way where you can contribute to research. And many of you have done and have done so very generously, and we're very grateful for that. So I will leave you with this last quote that I find to be a very powerful one. If you think research is expensive, try disease. So I hope I've relayed that to you, that my team and all the other scleroderma teams in the country and even in other countries are working very hard at identifying the causes of scleroderma and coming up with a cure for it. So finding a cure is not a mission impossible. It's just a matter of when, which day, basically. So I want you to remain optimistic about it. We will get there. All right, thank you for your attention. I'm going to move straight to the Jeopardy. I don't want you to be late for your other sessions, so let me do that. All right, and we're going to do this, typical of a Jeopardy game. Our staff, the Scleroderma Foundation staff, is going to help with this, because what we're going to try and do, it'll be hard for me to see who raised their hand first, or a paper, whatever you have, raise it if you have the answer, if we do the whole room at once. So what I would like to do is do section by section. So only the section we point to will be allowed to answer that question, and then we'll move on and on, section through section. The prize, here's the incentive. Earbuds in a nice Scleroderma Foundation case. So I hope you paid attention. It's too late now. Regrets will not help you. Let's start on that side. All right.
Are you ready, team? Okay.
So if you know the answer, lift up your hand or a piece of paper or something really quick, and we'll zero in on who lifted first, and then you'll get to answer it, okay? And to start off, I'm going to ask Mary, please pick a category and a dollar amount. And remember, your answers have to be in the form of what? You got it. I'm sorry? All right, she picked quotes for 300. Get ready. This person said, if you think research is expensive, try disease. Back there. You got it. Mary Woodard Lasker. Oops. Oh, bad. We'll make her say it again. All right. And then I'm going to ask you to pick the next category, please, and the dollar amount. Take a chance for 300. This is the section that gets to answer next, okay? Take a chance, 300. E4 is being made in this type of plant. The lady right there. Correct. What is tobacco? All right, you get to pick the next category, please, and amount.
Let's see. How about discoveries, 300?
Discoveries for 300. You are answering. Be ready. This animal is commonly used to study scleroderma. Oh my God, a bunch of
In the
ones.
blue, right here.
Robert got it.
What are mice?
Exactly. Very good.
Here you are. Your bites. And you want to pick a category and a dollar amount?
This and that for 400, please.
This and that, we're aiming high, 400. Okay. Last section, by the wall. You're it. The death of cows led to the discovery of this drug.
Warfarin. What is warfarin?
Close enough. Warfarin. We'll take it. All right. Could you please pick the next category and dollar amount, ma'am?
Oh, this and that for 300.
This and that for 300. We're going back. Section number 1. You guys ready? Ready. 300, right? My mouse froze. Okay. The drug used for this parasitic disease may be effective for treating multiple myeloma. Right there, lady in green.
What is Viagra?
No, the lady behind in the black top.
What is malaria? Malaria? What is malaria?
Anti-malarial. That's okay. Drugs for malaria, yes. Could you please pick the next category?
Okay. Fibrosis for 500.
Whoa. All right. In fibrosis, this section, fibrosis research, we measure levels of this extracellular matrix protein.
What is collagen?
Correct, absolutely. Thank you. All right.
Oh, thank you.
Whoops. That wasn't good. All right. Next category.
Take a chance 500.
Take a chance, 500. E4 is derived from this protein, this section.
Paying attention.
What is endostatin?
That'll work. What is endostatin, which is a piece of collagen 18? You're right, correct. The last section, could you please pick the category for the quotes for 200? This is for that last group over there. This person said the great use of life is to spend it on something that will outlast it.
Who is Pasteur? No? Okay.
Good try.
Nice try. He'll come up later. Oh,
Who is somebody Adams?
I'll take it.
Very good. Alex would have taken that.
We're flexible.
Take a chance 400.
Take a chance, 400. We're going back to this group. You guys ready? Anesthesia was discovered when it was noticed that this gas inhibited pain in people.
What is laughing gas?
Correct.
Discoveries 500.
Discoveries 500 for this section. You guys ready? Insulin was discovered when flies swarmed the urine of this animal.
Dog?
Correct.
What is a dog?
Correct.
Let's do quotes for 400.
This person said, the seeds of great discoveries are constantly floating around us.
Who is Plato?
Who is Joseph Henry?
Oh,
Perfect.
very good.
The quotes for 500.
Quotes for
500.
500. All right. This person said there are three kinds of people: those who let it happen, those who make it happen, those who wonder what happened.
Who is Richardson?
We'll take that.
Very good. And the next category, sorry.
Quotes for what's ever left.
Sorry. Quotes for
100.
100. This person said, chance favors the prepared mind.
Louis Pasteur. Or who is
Ah,
Louis Pasteur?
yeah, okay. Correct.
Discoveries for 400.
Discoveries for 400. This section next. This chemotherapy drug is used to cause fibrosis in mice. The lady in the gray.
Is bleomycin?
Correct. What is bleomycin?
Fibrosis for 400.
Fibrosis can affect this organ.
What is any organ?
I'm sorry?
Any organ? What is any organ? Do you want me to name them all?
Can we do two answers? the right there.
Right here.
All right. So actually, any organ is okay, although we were looking for a specific organ, but the lady right here also answered whether skin or lung or any of these other organs as well. All right, we're gonna let you pick the next category, and then it will be for that section.
This and that for 500.
This and that for 500. The discovery of this alcohol treatment medicine was accidental. The lady back there had her hand up already.
What is antabuse?
Correct?
Take a chance 100.
Take a chance 100. We're going to go back over there. I'm going to give Ryan a chance to catch his breath. And Ann is there. Okay, this and that 100, right? This drug for ED, which stands for erectile dysfunction, was discovered while treating patients with chest pain. I saw hands.
What is Viagra?
Correct. That was an easy one. All right, next category, please, for this section.
Discoveries 200.
Discoveries 200. This person discovered penicillin, the lady in pink right here.
Thanks, Ryan.
No. Anyone else from this section want to try?
Who is Fleming?
Who is Fleming? Correct. You were really paying attention. All right, next, please.
Fibrosis for 100, please.
Fibrosis for 100. For this section, this Latin word means outside the body.
Ex vivo? What is ex vivo?
That'll work.
Yeah. All right, guys.
This and that for 100.
Okay. This and that for 100 is done. There's 200 if you'd like. Would that work? All right, this section. This antibiotic was discovered when a fungus grew on a plate. I think
It was.
you saw.
I think it was.
Yep.
You were first, actually.
What is penicillin?
Yes, correct.
Congratulations, very good. And the next category?
Fibrosis for 200.
Fibrosis for 200. Fibrosis, we're going to go back section there. Fibrosis results from increased production of this protein.
What is collagen?
Correct. At least that's one of the answers, yeah.
Take a chance 200.
Take a chance 200. This Latin word means in glass.
What is in vitro?
Correct.
Fibrosis 300.
Fibrosis, I'm sorry. 300 is all we have, okay. For this section. This Latin word means in a living organism.
Right here.
Yeah.
I thought she was first. Oh, well.
She did. You
I think
no?
you were first.
Did you change?
You want to?
Living organism. In vivo.
In a question?
What is in vivo?
Correct. What is in vivo?
And you want to pick the next guy?
All right, we have two more.
Discoveries 100.
Discoveries 100, section over there. An example of an accidental discovery for jaundiced infants is this.
What is natural sunlight?
What is light? Correct. All right, oops. One last one I think we have.
And give her the category too.
We have one less category. And so we are gonna, it's take a chance 100 is all that's left, so we're gonna go back to the first group. Fibrosis research is important because fibrosis kills this % of people in developed countries.
20%? What
No.
is 20%?
No? I don't know who. I like it.
All right.
She's a go-getter.
45. What is 45%?
Correct, absolutely. Alrighty, everybody, you've been a great crowd. It's always such a pleasure to be here every year and to get to see you. Enjoy the rest of the conference. I wanted to say a big thank you to Carrie Conley, who's the conference director, who, as you realize, has done an amazing job scheduling topics that are of use to you and leading the team in organizing this effort. And thank you to Anne and Ryan and Robert for helping with the prizes. Have a great day.